I believe now it's working. Hi guys, am I audible to all of you? Oh, am I audible to all of you? I believe, okay, finally. Hi everyone, am I audible? Am I audible? Yes, please. Okay, finally. So, guys, basically, no excuses, but yeah, it's a fact that currently I'm staying at a remote area, so power issues are too much, and you know that. All that things, so much of rainfall and everything. So my system, right, even in my live class, it happened today, like my system was completely shut down, right? So now power is there and I'm here in front of you. Okay, so I've just tell me, am I perfectly audible and visible to all of you? Am I perfectly audible and visible to all of you? Yes, everybody. Am I perfectly audible and visible to all of you? I believe it's a yes. Thank you, Serena. Thank you so much. So I was just thinking, I was literally very frustrated. I was thinking that I should move my class for tomorrow, right? So I'll take it tomorrow. Let's not take it today. But finally, it started. Okay, so I hope I'm perfectly audible. I hope you all are doing very well. So guys, welcome to the session, and now let's finish this. Let's finish this chapter, right? It's been so long, right? We are going slow, I know that, and we don't have classes every day, so that is why we are just stretching it a bit. But yeah, it is one of the most important chapters. Last time also, the questions were not there from this part. Usually we get 2 to 3 questions from this, 3 to 4 questions from this particular chapter. But next year, like for the NEET 2026, yes, we are expecting the questions from this part. So you guys can do one thing. You can just, watch it, like once we are done with the chapter, you can just start from the part 1. You can just increase the speed and you can revise this chapter. We will definitely come up with the our marathons as well, but this is the suggestion that I want to give you. Chalo. So now let's get started. Baki, you know that here in the description box, you have the links, and you have the links, right? You have the links for our batches as well. So if you want to be the part of my batch, in the description box there is a link. You can click on that link and you can be the part of you can join my batch. Okay, and trust me, you are going to love it. The classes are going amazingly. We have started the ecology. We are also completing that plant physiology part. So I have started this portion first, right? Because at the last, whenever we start these chapter, you are not able to give it sufficient amount of time, but now you will be able to. Okay, so let's continue this part. So today we have to start the cell wall. In the last class we discussed the cell membrane in detail. So today what are we going to discuss? Actually, we are going to discuss something happened. Yeah, so we are going to discuss the cell wall. So let's start with the basic introduction. And as I said, I'll share the PDF with all of you guys, right? I'll share the PDF with all of you. And in that particular PDF, you will have all the notes point by point. Okay, so at this point of time, just focus on one thing, and that is understanding the topic, right? Just focus on one thing, that you have to understand the topic. That is the most important thing, right? That is the most important thing. Are you getting my point? Right? So you have to understand this particular topic. Now, cell wall. Where do we see cell wall? Definitely in the case of prokaryotes, we see the cell wall, yes or no? Yes or no? Right? In the case of, if I talk about the eukaryotes, so in the case of eukaryotes, guys, you are going to see the cell wall in some of the protista. I'm not saying all of the protista. In some of the protistans, you are going to see the cell wall. This is the one thing. Then, other than that, what is going to, where will you see the protistans? In the fungi, right? Other than that, where will you see the cell wall? In the fungi and in the plant cell as well. But if I talk about the animal cell, so do we have the cell wall in the animal cell? No, we do not have that, right? We do not have that. So let's discuss about that. So already you know that in the case of bacteria, when we talk about the cell wall, so bacterial cell wall, it is made up of, yes, I want to see the answers in the chat section. Yes, everybody. I want to see the answers in the chat section. Yes, everybody. Yes. Yes. Everybody, tell me. right? Bacterial cell wall is made up of, guys, I need the answer. It is made up of peptidoglycans. It is made up of what? Peptidoglycans. What else you call them? You call them as murines, right? You call them as murines. You can also call them mucopeptide. So when it comes to the peptidoglycan, and not just the peptidoglycan, of course, we are going to see some other amino acids, right? We are going to see some other proteins as well here in the cell wall, right, students? So now when we are talking about these peptidoglycans, yes, when we are talking about these peptidoglycans, so basically what is the point here? These peptidoglycans, they include NAM, NAG. What do they include? NAM, NAG. We have already covered this portion in the bacterial cell. So when we are talking about the NAM, NAG, so you will discuss about this thing, N-acetyl muramic acid, right? That's what we have. And we have N-acetyl glucosamine, N-acetyl muramic acid. And what else do we have? We have N-acetyl glucosamine. So N-acetyl muramic acid and N-acetyl glucosamine, that is something that is forming the bacterial cell wall. It is something which is forming what? Which is forming the bacterial cell wall. Now, if I talk about the fungi, do they have cell wall? Yeah, definitely. In the fungi also, we have the cell wall. So when you talk about that, that cell wall of fungi is made up of chitin and some other polysaccharides. What is it made up of? Which one is the main component in the cell wall of your fungi? It is going to be chitin. It is going to be chitin. So chitin and some other polysaccharides are there. So when we are talking about the chitin, so what exactly is the chitin? Chitin is also known as fungal cellulose, my dear students, right? And chitin is nothing, but chitin is also a polysaccharide. And what sort of polysaccharide it is? It's a homopolysaccharide. And what do we understand with the word homopolysaccharide? So homo means, students, same. So chitin is made up of same sort of, right, same types of monosaccharides. So many monosaccharides, they will join, same type of monosaccharide, or I would say same monosaccharide will join, and it is going to form the chitin. So chitin is also a polysaccharide. So when you are talking about the fungi, when you are talking about the fungal cell wall, so as I said, it is made up of chitin and some other polysaccharide. So when it is the chitin, it is also known as fungal cellulose. So as I said, it is made up of same type of monosaccharides. And what is that monosaccharide? That single subunit of chitin is your NAG, N-acetyl glucosamine. So many N-acetyl glucosamines are going to join. They will form the cell wall. In the case of your fungi, they are going to form the chitin, right? So other than chitin and some other polysaccharide, there are some proteins also which are present in the fungal cell wall. Okay, so the next thing that we have to discuss here is the plant cell wall, right? What we have to discuss here? We have to discuss the plant cell wall. So when you talk about the plant cell wall contains what? It contains cellulose, it contains hemicellulose. So cellulose, hemicellulose, what exactly are they? They are also the polysaccharide pectins and proteins. That's what you are going to see in the plant cell wall, right? You are going to see the cellulose, you are going to see the hemicellulose, you are going to see the pectin, you are going to see the proteins. Where? In the plant cell wall. And here, if I have to be specific, if I talk about algae, no doubt it's a plant, but when you talk about algae, in the case of algae, we have cellulose, we have galactins, we have mannans, right? We have cellulose, we have galactins, we have mannans, and minerals like calcium carbonate, they are also found in the cell wall of algae. So that's something you have to remember. Now, in the case of plant cell, when you look at a plant cell, okay, no doubt cell membrane is there, students. And then, if here I'll take two examples. Let's suppose you are talking about a plant cell, the plant cell which is immature, which is active, which is small sized, right? The plant cell which is active, which is small sized, which is immature, right? What am I talking? I am talking about the plant cell which is active, which is small sized, which is immature. So when I talk about such sort of plant cells, what will I see? I see that they have the plasma membrane. They have the plasma membrane, and they are having, right, they are having primary cell wall. Yes, in the case of plants, when we talk about the cell wall, we'll see primary, secondary, and tertiary. So here, what exactly are you going to see? You will see the primary cell wall, because the cell is small in size. So when we are saying the cell is small in size, when we are saying it is thin wall, when we are saying that this cell is, growing, dividing, growing, so you will see that such plant cells, they will have primary cell wall only. They don't have the secondary. Yes, here we will discuss primary, we will discuss secondary, we will also discuss tertiary, but here you people are going to see what? You people are going to see the primary cell wall. So this primary cell wall, it contains cellulose mainly. It is not very thick. It is thin walled. It is thin walled. It is elastic in nature, my dear students. Right? It is thin walled, it is elastic in nature. Because of this cell wall, the plant cell is able to expand. The plant cell is able to grow. That is what we see here in the case of primary cell wall. Right? That is what we see here in the case of primary cell wall. Right, so primary cell wall is thin walled, primary cell wall is elastic in nature, primary cell wall is helping in the expansion, it is helping in the growth of that particular cell, because this cell is still immature, it is growing. Right, it is expanding, it is elongating. Okay, so here what am I going to see? Primary cell wall. I'll come to the composition part. Then when you talk about a cell which is mature, right, which is mature plant cell, there what you are going to see? See, just imagine this is your plant cell. It is having the cell membrane. Now students, that's something you have to understand, right? When a cell is mature, right, when it has lost the ability to divide, when it has become a mature cell, then no doubt we will have the primary cell wall. Indeed, we will have the primary cell wall, but we will also have the secondary cell wall, but that secondary cell wall will present inner to the primary cell wall. Where will it present? It will present inner to the primary cell wall, and it will be in the form of layers. Let me elaborate. So what exactly I'm trying to tell you? Let's say this is a cell. This is having cell membrane. This is having cell membrane. And then after that, outside it, you have primary cell wall. Outside it, you have primary cell wall. Now, here you will see cell membrane. So here, of course, you will see primary cell wall, but when a cell, here I'm saying the cell is immature, here I'm saying cell is growing. So cell membrane, primary cell wall. And when your cell is mature, it will have secondary cell wall. So that secondary cell wall, my dear students, it is not, usually we imagine it like this, cell membrane, primary and secondary. No, it's not like that. Your primary cell wall will be towards the outer side. So inner to it, you guys will see secondary cell wall, and that too in the form of layers. That too in the form of layers. That is what we have to understand. Okay, the composition is going to be like this only: cellulose, hemicellulose, pectin. No doubt in the secondary cell wall, in different types of cell, the thickening will vary, the composition will vary. In some cases you will have cellulose, hemicellulose, pectin, along with that suberin, lignin can also be there. So that is the thing that varies, and that is something we will discuss. But right now you have to focus on this part, you have to understand it. If you guys read the NCERT, you guys will see that in the case of cell wall, it is mentioned that it is non-living rigid structure, right? I hope you all can see that. We discussed that cell wall is a non-living rigid structure, isn't it? But my dear students, when your cell is immature, when it is actively dividing, when it is growing, when it is immature, in the immature cell, you just have primary cell wall, and primary cell wall is considered as living. It is not considered as dead. It is considered as living. Once the cell becomes mature, once it gets these layers, then you will call it as what? Then you people are going to call the cell wall as dead. But this is the first thing that you have to understand. So if my primary cell wall, if it is thin, and if it is thin and living, if it is elastic in nature, so how this primary cell wall will grow? So in the case of primary cell wall, the growth will be via intussusception. What is going to happen here? Intussusception. So how many of you know the meaning of intussusception? Yes? How many of you know the meaning of intussusception? How many of you know the meaning of intussusception? Quick, everybody. Yes? Guys, intussusception, from in you have to remember the word internal. What is the word that you have to remember from this in? You have to remember the word internal. What is the word that you need to remember here, guys? You need to remember the word internal here. So when a structure is going internally, like you take our example, in our case, it's not like that, just take the example of a baby. That baby is growing. That growth is internal. The bones are elongating, right? The height is increasing. It's not like that on that baby, you will have multiple layers. It's not going to happen, right? That baby will not have multiple layers, one after another, one after another, one after another, then that baby will grow. No, the growth is internal. So that is the kind of growth we see in the living beings. So yes, in the case of primary cell wall, because it is living, it also shows the growth which is known as intussusception. So intussusception simply means internal growth, that this primary cell wall is growing internally. Now, as you know, we have just discussed that secondary cell wall is considered as, right? Your secondary cell wall is considered as dead. Your secondary cell wall is considered as dead. Right? It is considered as what? It is considered as dead. So because it is considered as dead, so here you will see that no doubt the cell is growing. No doubt in that cell that layers will form. But how? There is the accumulation. There is an accumulation, or I would say deposition. So what does it mean? You can compare it with the growth of the non-living beings. Like, you consider the example of a sand dune. In the case of a sand dune, that sand dune will also grow. But how? How? When the layers will get accumulated over it. When the layers will get deposited over it. Right? So this is the kind of growth you see in the non-living beings. Okay, so here in the secondary cell, here in the case of the secondary cell wall also, you will see that the layers are formed, right? That the layers are formed. So this type of growth is known as, my dear students, A for accretion. What is it, students? It is known as accretion. It is known as what? Accretion. So from this accretion, you people can remember the word accumulation. What is the word that you all can keep in your mind? You can keep the word accumulation in your mind, that basically the accumulation of material will occur. That's why the layers are there. That's why the layers are there. So that is your secondary cell wall. Is that clear? Now let's suppose, if by any chance, right? If by any chance there will be tertiary cell wall. So of course, firstly you will say primary plasma membrane. Then let's suppose it is your primary cell wall, right? Then below that, you people will see the layers of secondary cell wall like S1, S2, S3, and next to it, my dear students, you people are going to see tertiary cell wall in the layers like this. So where do we see tertiary cell wall? Yes. Where do we see tertiary cell wall? We see tertiary cell wall in the case of, some gymnosperms. In the tracheids of some gymnosperms, yes, in the tracheids of some gymnosperms, what you people are going to see? In the tracheids of some gymnosperms, what you people are going to see? You people are going to see what? You people are going to see the layers, right? Tertiary layer as well. What will you see? Tertiary layer. So if somebody is going to ask you about the sequence, which they can ask definitely, so of course, outermost is going to be primary cell wall. As per this I'm talking about. Then S1, S2, S3, then comes the tertiary. Then comes the tertiary, right? Then comes the tertiary. Is that clear? Any doubt? Any doubt? Sure? Okay. Now, next thing. Now plants. Like let's suppose this is one plant cell. Right? Let's suppose this is one plant cell. Right? This is another plant cell. This is another plant cell. This is another plant cell. Let's suppose the things are like this. Right? Let's suppose the things are like this. Now, here what is happening? what is happening here? 1 2 3 4, this is what we have. Now here what you people are going to see, right? Let's suppose this is your primary cell wall. Let's suppose these are the layers that I'm making. Let's consider it as secondary cell wall. Let's consider it as secondary cell wall. Just consider it like this. Now in between these two, right? Or overall, if I have to talk about the plant cells, in the case of plant cells, right? In the case of plant cells, my dear students, obviously you're talking about a multicellular structure right now. So you people will see that one cell is attached to the another cell. Yes, one cell will be attached to the another cell with a layer, with a cementing layer, right? With a cementing layer. What do we call it? Anybody? What do we call it? We call it as middle lamella. What do we call it? We call it as middle lamella. Middle lamella. So what is this middle lamella here? What is the purpose of it? Let me discuss that with you all. Just allow me 1 minute. So this middle lamella is basically the cementing layer, my dear students, right? This middle lamella is basically the cementing layer, my dear students. And this cementing layer, it is made up of calcium and magnesium pectate. Calcium and magnesium pectate, that is going to form this middle lamella. That is going to form this middle lamella, right? That is, yes, everybody please repeat it with me. That is going to form what? That is going to form middle lamella, isn't it? Isn't it? Isn't it? Yes or no? Yes, everybody, yes or no? If there is any doubt, do let me know here. So that middle lamella is made up of calcium and magnesium pectate. Am I right? But the main point here is that basically calcium and magnesium pectate is there, but more is calcium. So in NCERT also, it is mentioned that it is the calcium pectate that makes the middle lamella, right? So in the NCERT also it is mentioned that it is the calcium pectate that makes the middle lamella because the amount of calcium is more. So it is just like a glue. It is gluing the cells together. It is joining these cells together. So this is the middle lamella, cementing layer made up of calcium and magnesium pectate. And let me tell you, middle lamella, it develops from cell plate. Now you must be thinking, if you are doing this chapter for the first time, that what is cell plate? Cell plate is a structure that will form when the plant cells, they are dividing. At the time of their cytoplasmic division, in between there is a formation of cell plate, and that cell plate further represents this middle lamella. That is something we have to remember. We'll start the cell cycle and cell division, then you will be able to understand it. Now this is the middle layer. So this cementing layer or middle layer, it is absent on outer exposed part. It is not present on the outer exposed part. It is absent there on the outer exposed part of the plant cell. Okay, now in the case of this, right, you will see that in the plant cells there is a kind of structure which is traversing these layers, right? There is a kind of structure which is traversing this layer. What is the meaning of this traversing, or a kind of transmembrane structure? What is the point actually? Yes, what is the point actually? Actually, here in the case of plant cells, you people will see that even in the case of plant cells, there are continuous cytoplasmic connections. In the case of plant cells, what do we see? Yes, everybody, what do we see? In the case of plant cells, you people are going to see that there will be continuous cytoplasmic connections. What will be there, people? There will be continuous cytoplasmic connection. So what do we call them? We call them as plasmodesmata. What do we call them, students? We call them as plasmodesmata, the continuous cytoplasmic connection in between the plant cell, and that plasmodesmata, students, that is traversing from these, from these layers as well. And here in this plasmodesmata, you people are going to see, right, you people are going to see some structures which you are calling as desmotubules, right? desmotubules. It is just representing the endoplasmic reticulum there. So that is something we need to keep in our mind. So this is about the cell wall. I'll discuss the structure with you all. So here you are going to see that in the case of the cell wall, you people are going to see that when you are talking about the primary cell wall, you'll see how that cellulose microfibrils will form. That microfibrils are arranged, parallelly arranged, compactly arranged, then they form the structure. In between, we see the pectins. In between, we see the proteins. So that's how they form a stabilized structure, right? That's how they form the stabilized structure, isn't it? Isn't it? Are you getting my point? So this is what we have discussed about the structure of cell wall. Now, if I have to discuss the points and all, so let me tell you, for the very first time, who observed it? Robert Hooke. So he was the one who observed the cell wall first. Are you getting my point? He was the one who observed the cell wall first. Why? Because if you remember, he was the one who discovered the cell, who coined the term cell. So the cell that he saw was the dead cell. So actually under the microscope, he saw these walls only. So he was the first one who observed the cell wall. So now let's have a quick revision with the help of diagrams also, then we will revise it. So you guys can see the structures here, right? You people can just have a look of these structures. So plant cell wall, see the way they are attached to each other. Okay, so remember one thing, students. It is also a previous year question. If somebody will ask you which one is the outermost layer in the plant cell, middle lamella. Then you will have primary cell wall. Then you will have layers of secondary cell wall. Then you will have layers of secondary cell wall. So that's how it is present. That's how it is present. Are you getting my point? Okay, so here you guys can see this is the cell, outer middle lamella, then primary wall. So it is thin, it is living. But in the case of secondary, 1, 2, 3. So we have S1, S2, S3, and in between we have the lumen. So that's something you have to keep in your mind. And here you guys can see you have the cellulose. You will see that how this glucose will join. You will see that how this glucose is going to form what? It is going to form the microfibril, right? Like cellulose microfibril. Because so many glucose will join, they will form these cellulose microfibrils. So when they will form these cellulose microfibrils, right, they all will parallelly arrange. They are going to form this structure there. So you guys can see the arrangement, cellulose. Right, individually this yellow is the cellulose microfibril. So this cellulose is also a polysaccharide. So what is it made up of? Right, what is it made up of? Glucose only. Cellulose is also a polysaccharide of glucose. Cellulose is also made up of glucose only. Is that clear, everybody? Yes. So here you guys can see cellulose. You people can see this, bluish colored hemicellulose is there. In between, lignins are also there in some cells. So this secondary cell wall composition and thickness, it varies from cell to cell. It depends upon the function. So you see the pectin in between. You see the arrangement here. Okay, guys? You see the arrangement here. Now see, in some cases, of course, lignin is there. Cellulose, hemicellulose, pectin, right? So cellulose is your unbranched polysaccharide. Let me complete the biomolecules, then we will relate it there. Fungal cell wall made up of chitin, like I said. Bacterial cell wall made up of peptidoglycan. So this is important. All the main composition part is important here, right? It is important here. So you people can see this. Peptidoglycan will also include NAM and NAG. Peptidoglycan will also include NAM and NAG. Is that clear, everybody? Is that clear? And here you guys can see abundant this cytoplasmic connection, right? This is plasmodesmata. So P for plant and P for plasmodesmata. So as I said, it is what? It is nothing but the continuous cytoplasmic connection in between the plant cells. It is the continuous cytoplasmic connection in between the plant cell. Is that clear? So here, as I said, what you people are going to see? You people are going to see desmotubules. What you people are going to see? Desmotubules here. Desmotubules, that is basically your ER only. So here you have the points as well. Let's have a quick revision of that, and trust me, you will here in this live class only, you will be able to understand each and everything, and you will be able to understand, you will be able to revise that. So cell wall was first observed by, as I said, Robert Hooke. Okay, so bacteria, plant cell, fungal cell, they all are having cell wall, right? So fungal cell contains chitin. Again, this is the NEET PYQ. Right, this is the NEET PYQ, chitin. So chitin and polysaccharides are there. So this chitin, it is also the second most abundant. It is also the second most abundant organic molecule on Earth. It is the second most abundant organic molecule on Earth. Are you getting my point? It is what? It is the second most abundant organic molecule on earth, right? Which one? Your chitin. The first most abundant organic molecule will be your cellulose. Is that clear? Right? Is that clear? Okay, so the next point, algal cell wall. It contains cellulose, galactans, mannans. What exactly are these galactans and mannans? They are also the polysaccharides, just like cellulose. They are also the polysaccharide, and the minerals like calcium carbonate is there. So plant cell wall is having cellulose, hemicellulose, pectin, protein. So this is what I have already discussed with you all. Is that clear? Right? So other than that, when you look at the primary cell wall, right? Other than that, students, when you look at the primary cell wall, so you guys will see that it is found in meristematic cells. What are meristematic cells? What are meristematic cells? So these are the actively dividing cells. So just now I told you that if the cells are actively dividing, right, it means they are going to be immature. It means they are going to be active. And you know that the cell wall then is thin. The cell wall then is thin. Is that clear? So your meristematic cell, students, and your parenchymatous cells, they have primary cell wall. Am I clear here? Am I clear here? So where do you see primary cell wall in plant cell? You people are going to see it in the case of meristematic tissue. You people are going to see it in the case of parenchymatous cells. Am I clear? Am I clear? Next point. So young plant cells, they have single layer of primary cell wall. It is thin, elastic, capable of expansion. It support that living cell. Already discussed. That's how we revise. Okay, so primary cell wall will show internal growth, and we use the word intussusception for that. Right? We use the word intussusception for that. Next, secondary cell wall. So students, in mature cells, you will have more cell wall material will be added. But what is the point which we need to focus? That it will be added inner to the primary cell wall. So yes, this statement can easily give you an MCQ. So primary cell wall, then inner to it you will have the layers of secondary cell wall. Primary cell wall is single, primary cell wall is living, primary cell wall is having cellulose, primary cell wall is thin walled, it is not that thick, but the inner to that cellulose will be secondary cell wall, S1, S2, S3, like this you are going to see. Okay, so growth, as I said, is due to accretion or accumulation. Growth, as I said, is due to accretion or accumulation. Next, it is thick, it is rigid. Next, your cellulose, which is made up of glucose. So many glucose, they join, they will form cellulose. So cellulose microfibrils, they will be compactly arranged here. And in the matrix, we also see lignin and suberin. In the matrix, we also see lignin and the suberin. Okay, so this cell wall formation is not uniform in all the cells, as I said. So tertiary cell wall, I told you, tracheates of gymnosperms, they have the layers inner to the secondary cell wall. This will be tertiary. Then students, we discussed about what? We discussed about the middle lamella. Here I'm going to discuss few more important points. Middle lamella. So as I said, adjacent cells in a plant tissue, they are held together by a thin, sticky, amorphous cementing layer, middle lamella. Again, important MCQ. What does it contains? Calcium and magnesium pectate. Again, important MCQ. But which one is more? Here, calcium pectate. What is written in our NCERT? Calcium pectate. So yeah, this is something you have to note down here. Right? This is something you have to note down here. Right? So it is not present on. This middle lamella is absent on the outer exposed part, and it forms during cytokinesis. Okay, beta. So when your fruits, when they get ripened, they become soft now. How is it possible? The pectates present here, they will solubilize, they will form jelly-like material, and that is why your fruit will become soft. Is that clear? That is why your fruit will become soft. Next, this question, right? So those who are targeting 360 out of 360, right, especially after the level of NEET 2025, do not forget to mention such kind of things. So next thing is cellulose synthase. Next thing is cellulose synthase. Next thing is what, beta? Cellulose synthase. So this is actually an enzyme. See students, overall what do we discuss? We say that it is the Golgi apparatus that forms the cell wall. It is the Golgi apparatus that form all the content of cell wall. That is again your important MCQ. right? This is again your important MCQ, that it is the Golgi apparatus that is going to form what? That is going to form the cell wall. But the main thing in plant cell wall is cellulose. And how that cellulose is synthesized? It is synthesized by cellulose synthase complex enzyme. It is synthesized by cellulose synthase complex enzyme. So this enzyme is located in the plasma membrane, my dear students. So that is why, guys, that is why, yes, everybody, please focus here. That is why in plants, the cell wall is synthesized by Golgi apparatus, but when it comes to the cellulose, that will be synthesized by cellulose synthase complex, which is located on plasma membrane. So that is something you have to remember in your mention in your notes, and that is something you have to remember. Is that clear? Is that clear, everybody? So just have a look, right? Can you see these plant cells? Can you see these some, transmembrane structures? So these are basically plasmodesmata. These are actually the plasmodesmatal connections or plasmodesmata. Okay, so now look at this. Look at this part when you see it in the cross section. So here in between, right, if you see this part, two cells are connected, right? So they are connected with the middle lamella. This is the adjacent cell wall, right? They are connected with the middle lamella. So these cell walls are also traversing. So you will see, what will you see here? You will see the desmotubules in between. You can see this part. Right, this is your middle lamella, the plasma membrane, the cell wall is there. So in between, you will see the desmotubules. So they are representing your endoplasmic reticulum. That's what you can remember. Okay, that's what you can remember. So up to this part, if there is any doubt, do let me know, because then we have to start endomembrane system. Yes. Yes, everybody. Done? Done? Sure? Yes? Okay, great. So the next thing that we have to discuss is endomembrane system. What is the next thing, students, that we have to discuss? Next thing that we have to discuss is endomembrane system, which is quite important for your NEET examination. Now, let's understand what exactly is this endomembrane system. You know that in the cell, right? You know it very well that in the cell, we have so many cell organelles. Am I right? In the cell, we have so many cell organelles, but you will see that some organelles, students, they work in a coordinated manner. They work together like a team, right? They work together like a team. So how come team? Just a minute. Wait, let me show you this part. Okay, so the point is that in the cell, normally what you see that organelles, they are doing their job. They are working, right? They are just doing their activities. Now what happened? In the case of cell, there are some organelles which work together in a coordinated manner. So when I'm saying they are working together in a coordinated manner, means they are working together like a team. Like the function of one is dependent on another. Just let's suppose, just imagine you are 3 to 4 friends. One person is doing that chopping thing. Another person is like, "Theek hai, I am going to cook." Another person is like, "Okay, I'm going to clean." Another person is like, "Okay, fine, I'm going to serve." So basically, you all are working together in a coordinated manner. You all are working together like a team, isn't it? Isn't it? You all are working together in a coordinated manner. You all are working together like a team. So in the cell also, we have some organelles, right? Those who are closely related in such a way that they are working together in a coordinated manner. So here, in this case, what do we have? We will, the first organelle that we have to discuss here is endoplasmic reticulum. Next is Golgi apparatus. Then you will talk about the lysosome here. Then you will talk about the vacuole here. That's what you people are going to discuss. What you guys are going to discuss? You people are going to discuss endoplasmic reticulum. You are going to discuss Golgi apparatus. You are going to discuss lysosome. You are going to discuss vacuole. I'm repeating this part again, guys. What are you going to discuss? Endoplasmic reticulum, Golgi apparatus, lysosome, and vacuoles. So they are making your endomembrane system. Right, their functions are coordinated. Mitochondria, chloroplast, they are not the part here. They are basically, these are the organelles which we have here. Or even sometimes we also refer it as GERL complex, GERL, Golgi apparatus, endoplasmic reticulum, and lysosome. Right, so basically you are talking about this. Right, you are talking about this. You guys can see we have the nucleus, outer membrane of the nucleus. Yeah, right. We know that nucleus is also double membrane bound. So outer membrane of the nucleus is also, this is also extending, right? This is also extending. Yes or no? This is also extending like this, right? This is also extending like this. So outer membrane is in continuation with the rough endoplasmic reticulum. We have the Golgi apparatus here as well. So that's what we have to discuss. So we will start with the very first organelle here, and that is known as endoplasmic reticulum. Right, we are going to start with the very first organelle here, that is going to be endoplasmic reticulum. So students, if I talk about the endoplasmic reticulum word is associated with the network. How is it associated? It is associated with the network. When you are talking about the endoplasmic reticulum, it is also seen only under electron microscope. Let me elaborate this part to you. Actually, what is happening, no doubt, endoplasmic reticulum, it's quite, it is there in the cell, it is properly, grown up, the membrane and all, everything is there. Like it is like a network, it is present in the cell. But still we are not able to see it under the light microscope because its membrane is very, very, very thin. It's a single membrane bound organelle. Membrane is very, very, very thin. So we are able to see it under the electron microscope. Is that clear? Now, when you're talking about this endoplasmic reticulum, its main function, like it is having two different types. Accordingly, its functions are also there. But overall, if I talk about this organelle, very important it is, helping in protein synthesis, helping in lipid synthesis, right? Helping in detoxification as well, right? Helping in glycogen metabolism as well. So these are the functions of this endoplasmic reticulum that we have to discuss. So when we start this part, so as I have already discussed with you all, so endoplasmic reticulum is noticeable with the electron microscope only, number one. So it is present in almost all. So when we are saying almost all, means not in all, but yeah, majority of eukaryotic cells are having endoplasmic reticulum, but the cells like ova, my dear students, like embryonic cells in the mature RBC, of course, mature RBCs, they are enucleated, they don't have these organelles as well. So they do not have the endoplasmic reticulum, right? Who doesn't have the endoplasmic reticulum? Your ova, right? The mature mammalian RBC, the embryonic cells, they don't have the endoplasmic reticulum. And when it comes to their amount, of course, their amount varies from cell to cell, students. So in the muscles, we can call this endoplasmic reticulum as the sarcoplasmic reticulum. So before going forward, we need to discuss its three-dimensional structure, its physical structure first. Okay, whose? Of this endoplasmic reticulum. So when you talk about its physical structure, what exactly are you going to see? You will see cisternae, you will see tubules, and you are going to see vesicles, right? You are going to see vesicles. Check Ames paper, right? Check Ames PY. It's a Ames PYQ regarding its structure, right? So regarding the structure, once there was a question, not just from endoplasmic reticulum, but yeah, also from the Golgi apparatus in your Ames exam. Okay, it was there in your Ames exam. So we have three structures. We have cisternae, we have tubules, We have vesicles. So I believe in your class 9th, you have studied that when it comes to the endoplasmic reticulum, we have the smooth endoplasmic reticulum and we have the rough endoplasmic reticulum. When we are saying, students, rough endoplasmic reticulum, we are saying that on its outer surface, on its membrane, ribosomes are attached. We'll tell you why. Right? In the smooth endoplasmic reticulum, we do not have that. Now, when we are now we are going to start this physical structure. So now you have to focus which part can form the smooth endoplasmic reticulum and which part can form what? Rough endoplasmic reticulum. So let's have a look. So first word that we have here is cisternae. What is the first word that we have here, guys? We have the first word, and that first word is known as cisternae. Cisternae is what? It's a sac-like structure. What exactly it is? It's a sac-like structure, right? It's a sac-like structure. Yes or no? Yes or no? It's a sac-like structure, pouch-like structure, sac-like structure. It is what? Cisternae. Right? What? Cisternae. So cisternae is what? It's a sac-like structure, a kind of pouch-like structure which are stacked parallelly to each other. Like, let's suppose if I'm saying this is the structure, so it is going to be stacked like this. Right? It is going to be stacked like this. So another thing that we are going to observe here is that in these cisternae here, you have certain glycoproteins. Right? You have certain glycoproteins. What do you have, guys? You have certain glycoproteins. So when you people talk about that glycoproteins, you will have the examples of ribofurans 1 and ribofurans 2. Ribofurans 1 and ribofurans 2. So this is what we have. So what these glycoproteins are going to do? These glycoproteins are going to help in the attachment of ribosome. Right? So imagine if these glycoproteins are present, if these glycoproteins are helping in the attachment of ribosome, so can I not say that these cisternae are the one that are going to form my rough endoplasmic reticulum? Yes or no? That it is the one which is going to form my rough endoplasmic reticulum. Isn't it? Isn't it? So you guys can see here that cisternae, they are flat, they are unbranched, they are sac-like structures, they are arranged parallelly to each other. They are in the form of stacks, like 1, 2, 3, 4, in this way. Then they bear ribosome. So I told you, why are they going to bear ribosome? Because they contain the glycoproteins ribofurans 1 and ribofurans 2. Ribofurans 1 and ribofurans 2. So that glycoproteins will help in the arrangement of your attachment of your ribosome. So of course, if it is helping in the attachment of ribosome, so it will result in the formation of rough endoplasmic reticulum. It will help in the formation of what? It will help in the formation of your rough endoplasmic reticulum. Is that clear? It will help in the formation of what? In the formation of rough endoplasmic reticulum. That is what we have. That is what we have. So just imagine, if this is the cisternae, if it is forming rough endoplasmic reticulum, if it is having the ribosome, right? If it is having the ribosome, so what is the point here? You have to understand it, that this will help in the protein synthesis. How does it help, beta? It will. These rough endoplasmic reticulum is actively involved in the protein synthesis. So from this part only, you guys can keep it in your mind that obviously we will see it in such type of cells which are basically showing active protein synthesis. Who? Rough endoplasmic reticulum. Next is tubules. Tubules, of course, some tube-like structures, some irregularly shaped structures, right? Some branched structures they are. So they do not have ribofurans, so they majorly form, students, smooth endoplasmic reticulum. What will it form? Smooth endoplasmic reticulum. So let me remind you, this is again a previous year question, that when it comes to the smooth endoplasmic reticulum, who's going to make that? It is your smooth endoplasmic reticulum. It is your smooth endoplasmic reticulum. Then comes the vesicles. These are some, round, oval structures. These are some round, oval structures that we have here. Is that clear? Is that clear? So they are irregular branched tube-like structures. They occur at the periphery of endoplasmic reticulum. They may be connected with the cisternae. Then comes the vesicles, which are oval, my dear students, which are round sacs, my dear students. They are freely there in the cytoplasm. So it is the tubules, these are the vesicles that are without ribosomes. So majorly when you guys will see the smooth endoplasmic reticulums, then you people are going to find the tubules, you people are going to find the vesicles. So is there any doubt? Yes, everybody. Is there any doubt from the structure part? Do you have any doubt from the structure part? Anybody? Yes, please. Yes, please. I hope it's crystal clear to everybody. Okay, so I told you this thing as well, that endoplasmic reticulum may pass from one plant cell to another, right, through this plasmodesmata. And how? Here you will see it in the form of desmotubules. Okay. Okay, so now let's discuss about its function. So we will start with smooth endoplasmic reticulum. We will start with smooth endoplasmic reticulum. So students, if it is smooth, it doesn't have the ribosome. So it is not concerned with the protein synthesis, but it plays some very important roles. Like it is having very important role in the lipid synthesis. Right? In the lipid synthesis, it is having a very important role. So basically cholesterol, which is again the type of a derived lipid only, so that cholesterol will be synthesized by this. So cholesterol is again, what it is the type of steroid. So this point is explaining many PYQs, right? This point is explaining many PYQs. So your SCR is involved in the lipid metabolism. It is helpful in the lipid synthesis. It forms the steroid. It helps in the formation of steroid. It helps in the formation of cholesterol. So you know that steroidal hormones, the hormones which are having steroid in it. So of course we can say SCR can help there. SCR can help there. So your tubules and your vesicles, they form your SCR, right? So basically tube-like structure or rounded structure this SCR is having, right, students? So SCR is also a storehouse of calcium. What exactly it is? It is also a storehouse of calcium. So because it is a storehouse of calcium, it is having the calcium, so it releases it when required. So its major role is there in the muscle cells. Do you know that? Its major role is there in the muscle cells. So in the muscle cells, the smooth endoplasmic reticulum is known as sarcoplasmic reticulum. What do we call it, guys? We call it as sarcoplasmic reticulum, which is again the storehouse of calcium, and that calcium is very important for the muscle contraction. That calcium is very important for the muscle contraction. Is that clear? Is that clear? That calcium is very important for the muscle contraction. So that is the role of your SCR. Moreover, it helps in detoxification. How does it help? It helps in detoxification. So in your liver cells, you have smooth endoplasmic reticulum that helps in detoxification, that helps in metabolizing many drugs and all. Are you getting my point? So here the functions are very important. So let's have a quick revision of these functions. So when it comes to SCR, it is the site of glycogenolysis and gluconeogenesis. So what is the meaning of these words, ma'am? Students, the first word here is glycogenolysis. What's the meaning of lysis? Your lysis means breakdown. Your lysis means breakdown. Glycogenolysis. Whose breakdown will be there? Glycogen's breakdown. So of course, if we are going to break the glycogen, glycogen is made up of glucose only. It's a polymer of glucose. It's a polysaccharide of glucose. So of course, what's going to happen? Laptop. Guys, am I audible? Yes, please. Am I audible? My laptop. Mm. Now it's working fine. Actually, some issue was there in my laptop. Now I believe it's okay. It's fine, right? It's fine, right? Am I audible? Yes, please. Am I audible? Okay. Sure? Okay. So basically we have the right we have the glycogenolysis, breakdown of glycogen. So breakdown of glycogen will give me what? Will give me glucose. It will give me what? It will give me glucose. Again, gluconeogenesis. What's the word that we have? We have the word gluconeogenesis. So genesis means formation, my dear students. What is the meaning of genesis? Genesis means formation. Neo means new, right? Then glucose. So basically glucose new formation. What's the meaning of that? It simply means that your glucose will be formed from a new source, that is your non-carbohydrate source. So basically when in need in the body, when there is a requirement of the glucose, glucose can also be synthesized from a non-carbohydrate source. That is the gluconeogenesis, students. That is the gluconeogenesis. Are you getting my point? So it is the site of glycogenolysis or gluconeogenesis. As I said, storehouse of calcium. That's why it is known as sarcoplasmic reticulum in the case of muscles. So it doesn't have ribosome. So you will see the SCR is well developed in muscle cells, in the fat cells, in the spermatocytes, and the cells that synthesize steroids. So the steroid point is very important. Okay, this is very important. Then next, I told you about the detoxification, that in the liver cells, which organelle is helping in the detoxification? This is your SCR, right? Then SCR also produce your Golgi apparatus, right? Help in the formation of this in the lysosome and in the vacuoles. Is that clear? Is that clear? So the next point that we have is rough endoplasmic reticulum, that is your RER. So if it is having the ribosome, right? If it is having the ribosome, definitely it is going to synthesize the protein. It will be found in those cells which are, actively involved in the protein formation. So who's going to show its structure mainly? It is going to be cistern. It is going to be cistern. Is that clear? So it is found in liver cells, it is found in pancreatic cells, in fibroblast. Fibroblast, remember? They are the matrix producing cell in connective tissue. Goblet cells, right, that forms mucus. There also you will find it. So it doesn't have calcium, but yeah, when it comes to RER, it will help in the protein synthesis. Okay, so RER also produce enzyme precursors, right? So lysosomes, they contain the enzyme. So the precursor for that enzyme, precursor like the structure, the initial structure which you need to make something, right? So precursor. So precursor for the formation of lysosome is prepared by your RER only. So RER, when it will lose its ribosomes, it can also form SCR. So this is something we have to keep in your mind, keep in our mind. Okay. Okay, so proteins will be formed on the surface of the endoplasmic reticulum. That proteins will be passed here in the lumen, my dear students. So here they will be modified. Here, what will happen to them, my dear students? They will be modified. Here, they will be modified. Is that clear? Is that clear? So enzymes of endoplasmic reticulum, they use substrates from your cytosol. Cytosol means cytoplasm, and they are going to form new phospholipids as well. So this is all about the list of their functions. So if you know any other important function, right, so obviously you people can mention that in the comment section, isn't it, guys? You people can mention that in the comment section. Okay, so next is the Golgi apparatus. Next organelle that we have to discuss is Golgi apparatus, right? The name of this organelle is on the name of the name of its discoverer, Camillo Golgi. Okay, right? Golgi apparatus, a very interesting organelle. So this is visible under both light as well as electron microscope. Who? Your Golgi apparatus. So it is visible under both It is visible under both, be it the light microscope or it is the electron microscope, it is visible under both. Now, the next part, as I said, it is discovered by Camillo Golgi, scientist name, important it is. So in 1898, in the nerve cells of the barn owl, the commonly found owl, right? Owl, the Tyto, the scientific name is Tyto here. Right? I need to recheck it again, but as per right, currently I am able to recall. Yeah, that is Tyto. So in the barn owl of this nerve cell of barn owl and cat, you will see the nerve cell. So it is present in all eukaryotic cell, but yeah, it is absent in few types. Just for an example, you will not see it in mature mammalian RBC. In the mature mammalian RBC, Golgi apparatus is absent. Is it important? Yeah, it is important. In the sperm cells of bryophytes, right? Sperm cells of bryophytes, they don't have that. In the sieve tube of plants, which are the member of phloem, they don't have that. Prokaryotic cells, they don't have that. Okay. And in the case of invertebrate and plant cells, right, they usually have many Golgi complexes which are known as dictyosomes. So yeah, dictyosome is the term that we use for the Golgi complexes of the plants. Okay, so they are present throughout the cytoplasm. They are present throughout the cytoplasm. So now when you look at the structure that I have made, I thought I'll explain it there, but chalo, anyhow, I'll explain it here only. So guys, so make sure you just write down all the points. So when you talk about this particular structure, here also you will see they are stacked parallel to each other, isn't it? Can you see that, students? Can you see that here? That they are also stacked parallel to each other, isn't it? See this. So in the case of Golgi apparatus also, right, you will see Golgi apparatus or Golgi complex, you will see that cisternae, tubules, vesicles, they are present here. So when you talk about this particular structure, you are talking about the cisternae. What are you talking? You are talking about the cisternae. And when you are talking about these cisternae, you will see they are also parallelly arranged to each other. But what is the main point here? See, Golgi apparatus, it is the organelle with the polarity. I'll discuss that with you. I'll discuss it. See, you can see the two sides here. When you look at this side, it is the cis phase, it is the forming phase, it is the F phase, and it is also known as the convex phase. This side is towards nucleus, right? This side is towards nucleus. Basically, this particular organelle is having two different sides. This is the cis phase, also known as forming phase, F phase, convex phase, which is towards the nucleus. This side is known as the trans phase. It is known as the concave phase. It is known as the M phase, or we can also call it as maturing phase. Right, we can also call it as maturing phase, and it is also known as concave phase. So where is it? Where is it? So this one is towards plasma membrane. This is towards plasma membrane side. Right, this is towards plasma membrane side. So that is something you have to remember. Okay, that is something you have to remember. Are you getting my point? So the cytoplasm in which it is present, that cytoplasm is known as zone of exclusion. What exactly is this zone of exclusion? Right here, I am writing it also. Golgi apparatus present in the Golgi apparatus is present in the cytoplasm where no other cell organelle is found. You don't have any other cell organelle here. So that is why you are calling it as zone of exclusion. That is why you are calling it as zone of exclusion. What are you calling it? You are calling it as zone of exclusion, right? Now when you look at their ends, can you see the ends here, guys? Can you see the ends here? So can you see that these are the swollen ends? So we will just read about it. We will just see that how from these Golgi apparatus, guys, right, from these swollen ends, some vesicles will also form. This Golgi apparatus is responsible for the formation of lysosomes as well, right? So exactly what is happening. Let's suppose this is your endoplasmic reticulum. This endoplasmic reticulum has made a protein. From this endoplasmic reticulum, see, initially I didn't mention, I just mentioned you that they work together in a coordinated manner, but I didn't discuss the coordination here. But now I'm telling you, because now you will be able to understand it. Let's suppose this is your rough endoplasmic reticulum. Let's suppose it has made a protein. Now, from this rough endoplasmic reticulum, my dear students, that protein will be there in the transition vesicles. That protein will come in the transition vesicles or transfer vesicles, whatever you want to name it. So these are some rounded structure that will have the protein which were made here. Now this protein containing vesicles, they are going to fuse with this cis phase, right? They are going to fuse with the cis phase here, cis side here, right? This transition vesicle. If you think that how are they going to move, of course in the cytoplasm here we have the cytoskeletal element. What do we have here in the cell? We have the cytoskeletal element. That cytoskeletal element will help in the movement of these vesicles. That cytoskeletal element will push that vesicles, and then that vesicles will go, they will fuse with the cis phase. So now the protein which is present here, it will enter here in the lumen of the cisternae. It will get modified here. It will get modified here. If the carbohydrate needs to get attached, it will be done. So all the modifications will be done here in your Golgi apparatus. Are you getting my point? Are you getting my point? So that is why this Golgi apparatus is the main sorting hub. It is main sorting site. It will sort things here and there in the cell, that this thing I need to send here, this thing I need to send there. So that is why this Golgi apparatus is also known as the traffic police of cell, my dear students. That is why, what do you call it? You also call it as traffic police of cell. What do you call it, people? You also call it as traffic police of cell. Who? This, right? This, your Golgi apparatus. So from transition vesicles, things are coming here, they are getting modified from one cisternae to another, they will move, and from this side, from the maturing phase, you will see some secretory vesicles will come out. That secretory vesicles are having the final product. Right, that secretory vesicles are having the final product. So that secretory vesicles, if they have signal, like whatever is the final product, it's not mandatory that it will have protein only. Let's suppose it is having SER. SER is sending the lipid there. So that lipid will also get modified here. It depends what kind of product is there. So final product, that final product will have the signals attached to it, right? That whether we need to keep that product in the cell only, whether we need to give it to the other organelle, or we need to release it out the cell, or we need to do the exocytosis of it. So accordingly, secretory vesicle will carry it. So that is how they are working together in a coordinated manner. That is why we are saying that they are making the endomembrane system. Is that clear? That is why we are saying that they are making the endomembrane system, because that's how their function is in a coordinated way. So the packaging and the sorting of the things will be done here in the Golgi apparatus, right? The packaging and the sorting out of the things will be done here in the Golgi apparatus. Is that clear? Is that clear, everybody? Yes, is that clear? Are you sure? So that is how, so that is what will be done here. That is what will be done here. So am I clear? Yes, yes, please. Am I clear? So that will happen here. So from this side, your vesicles will come out. So this is it. This is it. So all that sorting, packaging will be done here. Modification of stuff will be done here, right? Endoplasmic reticulum might send the enzyme precursor. That enzyme precursor for the lysosome enzymes will get further modified there. So then from these swollen ends of the Golgi apparatus, lysosomes will also come out. So that's how it forms. That's how it forms the vesicle. So this is something we have to discuss. So glycoprotein formation, glycolipid formation, where will it be done? In the Golgi apparatus. So in simple words, glycosylation or glycosidation, this is the question for all of you. You have to mention it in the comment section. What is the meaning or what is the difference in glycosylation or sidation? So that will also be done here in the Golgi apparatus. Okay, so Golgi apparatus, internal reticular apparatus, this is its name. Export house is another name. traffic police of the cell is also its another name. Let's have a quick revision now. Then, zone of exclusion. So, Golgi is covered by the zone of cytoplasm where no other cell organelle is there. So, that's why students, we call it as zone of exclusion. Then, next is structure. I already told you cis phase is there, trans phase is there. So incoming transport vesicle. Rather than transition vesicle, you go for the term transport vesicle, right? Though it's a kind of transition only, the thing will pass from this to that. But yeah, more appropriately, you just write the word transport vesicle here, right? Transport vesicle here. So cisternae, they are forming. And yeah, this question was there regarding their structure was there in the aims. So composed of many flat disc-shaped sacs called cisternae. The size is near about 0.15 micrometer to 1 micrometer in diameter. This is stacked parallel to each other. So the number, it varies. As I said, it is curved, flat, and the ends are swollen, right? And the ends are swollen. So it is arranged concentrically, means having same center. Something which is having same center. So concentrically, they are arranged usually near the nucleus, students. So we have the cis phase, also known as convex phase, proximal phase, forming phase. You can also call it as F phase. So it is usually near nucleus and the endoplasmic reticulum. When it is trans phase, maturing phase, M phase, distal phase, right? This is proximal. So this one will be distal concave phase. It is directed towards the plasma membrane. So both the phases are entirely different, students, but still they are what? They are interconnected. Is that clear? Both the phases are entirely different, but still, what are they? They are interconnected. Is that clear? Is that clear, everybody? Is that clear? Okay, so Golgi apparatus, they don't have ribosomes, so that's why it looks like SCR. So tubules are also there, beta. Already I have discussed that they are towards the periphery, right? This is towards the maturing phase of the Golgi, right? This part, right? So they are also interconnected with the cisternae. Vesicle, we know that round structures, right? They will arise, they are attached to the tip of the tubules. They will have the secretory substance. So when it comes to the function of Golgi apparatus, very important is the packaging of material, right? So Golgi will modify, it will sort and pack the material which is coming from ER, okay? So even not just coming from ER, if something is getting synthesized in the Golgi apparatus, that will also be released, right? That will also be released. It will also be modified there. So Golgi apparatus will receive the material from ER. I told you already through its cis phase. So a transport vesicle from ER will come that will fuse with the cis phase, right? And the substance will be chemically modified here in the Golgi apparatus. So students, glycosylation or sidation, these are two different words. So glycosylation and sidation of proteins and lipid is also taking place. So what is the difference in these two words? That is your homework. You have to mention it in the chat section, okay? You have to mention it in the chat section, okay? So it will form glycoprotein, glycolipids. Then finally, materials will be formed. Done? Okay, so vesicle will pinch off from the trans phase of the Golgi. They'll be discharged outside the cell. So Golgi apparatus is also known as the director of macromolecular traffic in the cell. So that's what we have to remember. Rest students, Golgi apparatus, it also forms the lysosome. Important MCQ. It also forms cell wall material. But do not forget that your cellulose will be synthesized with cellulose synthase complex, and that enzyme, where is it present, students? Where that cellulose synthase complex is present, that is present on the plasma membrane. Then cell plate formation will be done. Acrosome, what is acrosome? It is in the human sperm. That will also be done with the help of Golgi apparatus. Okay. So next organelle that we have to discuss is your lysosome, right? Christian D. Dubé. He was the one who discovered it. So it's a single membrane bound organelle. What sort of organelle it is? It's a single membrane bound. I believe you all know that it is known as the suicidal bag of cell. We also call it as recycling center, my dear students, right? We also call it as disposal unit of the cell, right? So you call it as suicidal bag, recycling center, disposal center, everything to them. So it's a single membrane bound organelle, right? So Golgi apparatus, your endoplasmic reticulum and Golgi apparatus, together they are forming this lysosome, because some of its enzyme, the precursors are formed by then further will be modified here. So finally this lysosome will be developed. This lysosome will be developed, which is very, very, very important. So in the plant cell, you people are not going to see the lysosomes mainly, because in the plant cells, you people are going to see that there is a large central vacuole, my dear students. What is there in the plant cell? There is a large central vacuole. So that vacuole is also having some, hydrolytic enzymes that also behaves like lysosome. So in the plant cell, you people are not going to see that. Is that clear? In the plant cell, you people are not going to see that. So when we are talking about the lysosome, lysosomes are fully loaded with the hydrolytic enzymes. And these hydrolytic enzymes or these hydrolases, right, that will include your proteases, that will include your carbohydrases, that will include your lipases, etc., etc. So these are the enzymes which are present in your lysosome. These are the enzymes which are present in your lysosome. So when you people talk about the lysosome, so it's a single membrane bound organelle, right? And this is rich with sialic acid, right? The membrane here is also having the sialic acid. So that will also make sure that the enzymes present within, they should not harm this lysosome membrane. Though these enzymes which are present in the lysosome, students, they are inactive enzymes. They are not even active. They are inactive. Actually, for their activation, they need the pH approximately 5 pH to get activated, right? So they need pH around 5 to get activated. Otherwise, they are inactive only. So whenever we need their enzymes to work, right? So here in the lysosome membrane also, you will see some proton pumps, due to which H positive will be pumped here, right? The conditions here will become acidic and your enzymes will get activated. But normally you will see that enzymes are in inactive state. So around 50 or more than that acid hydrolases are present here in the lysosome. So due to these enzymes only, it is having the capability to digest the different food substrate. It is having the capability to digest or destroy the cell if it is not working well or any other organ. So that's something now that we have to discuss. Is that clear? That is something now that we have to discuss, that why do we call it as suicidal bags? Yeah, why do we call it like this? So as I said, the hit was discovered by Christian D. Dubé, right? So here you guys can see lysosome, single membrane bound, filled with hydrolytic enzymes. So it is also noticeable with only electron microscope. Christian D. Dubé in 1955 first reported it. So students, it is there in animal cells and in some protozoans, but again it lacks in mammalian RBC. So they are less prevalent in plant cell than in animal cell. In animal cell, they are too prevalent, right? Too much, their presence is there. They are the suicidal bags. They are much, much, much needed. But in plant cells, it's not like that, because in plant cells, as I said already, we have the large central vacuole that will behave like lysosome. Is that clear? That will behave like lysosome. So in fungi also, their number is too much. So cells that, engulf the foreign particles, your macrophages, your WBCs, and many secretory cells of pancreas, students, they have too much of lysosomes. Is that clear? So I already told you, contain hydrolytic enzymes, your acid hydrolases are there, and they need the acidic pH. So normally these enzymes are inactive. Normally these enzymes are inactive. So when they will get acidic pH, only then they will become activated. Okay. So now one another thing that we have to discuss here is, when it comes to the lysosome, like see, we discussed Golgi apparatus. We said Golgi apparatus is an organelle with the polarity. It is having two faces. It is having cis side. It is having trans side. Isn't it? That's what we discussed. Isn't it? Yes or no? Of course. Right. Of course. We have this. Yes or no? Of course, we have this. So now the point is what? So now the point here is what? The point here is that when, as I'm saying that Golgi apparatus is an organelle with polarity, so this is the organelle with polymorphism. It is having polymorphism. It is the one that shows polymorphism. So poly, what is the meaning of poly? Poly means many. Morph means form. So it is having different forms. Like let's suppose this is the freshly formed lysosome, newly formed lysosome, having inactive enzyme. So we will call it as primary lysosome. What are we going to call it? Primary lysosome, my dear students, because it is having the inactive enzymes. Right, freshly formed lysosome it is. Now, when with this lysosome, let's suppose there is a food vacuole, if it will get fixed here, if it will get fused here, like let's suppose this is your primary lysosome, this is your food vacuole, if they both will get fused, of course, the food stuff, all the enzymes, they all are mixed. So then you call it as secondary lysosome. Or we can also call it as heterophagosome. Or we can also call it as digestive vacuole. So that is how it is. So H positive will be pumped here. Enzymes will get activated. They are going to digest it. They are going to digest it. So now, after that, whatsoever is remaining, this will be your residual body, right? The third form of this lysosome. You can also call it as tertiary lysosome. So it's simple. Newly formed lysosome with inactive enzymes, primary, right? The lysosome, when with primary lysosome, when your food vacuole will get mixed, then it will form secondary lysosome, heterophagosome, digestive vacuole. Then after the digestion of all the food, when you have the vacuole here, when you have the lysosome here, when it is having the remaining food, when it is having the residual body, so it is the tertiary lysosome or the residual body. Are you getting my point? Are you getting my point? Now, let's suppose in a cell, you have this organelle. This organelle is not working well. The cell is like, obviously genes are there, proteins are there. So there will be a kind of signal. So lysosome will be like, I have to destroy it. So lysosome is going to engulf the cell organelle, is going to destroy it. So this is, students, known as autophagy, right? Yes, so this is basically known as what? Autophagy, that is the self-eating. Right, that is the self-eating, like some organelle or a part here is not working, the lysosome is going to destroy it. The lysosome is going to eat it up. Right, this is the one thing. This is the one thing that we have to discuss. So in that case, you people will see that some autophagosomes will form. Right, just if we use the word phagosome. We use the word phagosome. I have discussed that in the cell membrane as well. Right, like let's suppose when a cell is going to eat something, when there is something for the cell eating, vacuoles will form. We use the word phagosomes there. So autophagosomes, it is having something from that cell only. It is having some organelle which is not functioning well. So when that autophagosome will fuse with, can also fuse with this lysosome, the lysosome will destroy its content. And sometimes what happens, a cell is not working well, its functioning is improper, right? So there is a signal for the cell death that says this cell needs to get destroyed. This cell is not working well. We need to destroy this particular cell. Right, we need to do what? We need to destroy this particular cell. So in that case, what is going to happen? All the lysosomes here, they are getting the signal. They all will fuse. They all will pass their content in the cytoplasm. And that will result in destroying this complete cell. So that is when we call them as the suicidal bags. Is that clear, students? That is when we call them as the suicidal bags. Is that clear? Is that clear, guys? So this is something you need to keep in your mind. So let's have a quick revision once again here from this part. Primary lysosome, also known as storage granule. So they store enzymes in the inactive form. They are newly formed lysosomes. Then digestive vacuole or heterophagosome. Digestive vacuole or heterophagosome. They are formed by the fusion of primary lysosome and phagosome. So what is this phagosome? It's a kind of vacuole. If it is the food, if it is having food, you can call it as food vacuole. If it is having some organelles from the cell, the organelle which is not working, you can call it autophagosome. Then residual body, tertiary body, undigested material is there, which will be removed by exocytosis. So exocytosis means something will thrown out outside the cell. It is the cell vomiting. It is the cell vomiting. Then, students, we have the another term that is known as autophagic vacuoles. right? We have another term that is known as autophagic vacuoles. So auto means self, phagy, eating. Then what's going to happen here? Autophagosome, which is having the organelle or the part which is not working well. You can call it as cytolysosome. So when there is the fusion of primary lysosome with some old dead organelle, then it is forming this autophagic vacuole. Okay, so lysosome, that is containing cell organelle to be digested. So auto means self. Now it comes under that self-eating, right? Now it comes under that self-eating. Is that clear? Is that clear? Sure. Is that clear? Are you sure? Yes, everybody. Are you sure? Okay, so lysosome containing cell organelle to be digested are autophagosome. So heterophagy is, hetero means different. So when there is the digestion of foreign particle, right? Like let's suppose this is a cell, something is entering, some foreign particle, right? Obviously, it can be in the form of phagosome or pinosome, right? Matlab, it can be some solid or the liquid. It has entered the cell. Lysosome is destroying it. This is something which has entered the cell from outside. So we will call it as heterophagy. So what is autophagy? Self-eating. So digestion of old or dead cell organelle. That is autophagy. This is the difference in these two. Then students, lysosomes of bone-eating cells. Do you know bone-eating cells? Like in our bones also, we have these cells which will destroy the bones. Osteoclasts there. So they are, right? So they are the lysosomes of these cells. They can dissolve the unwanted parts of bones as well. Autolysis, when they will become the suicidal bags. So all the lysosomes of the cell, they will burst, they will dissolve that cell completely. So example will be reabsorption of the tail of tadpole is done by lysosome. Is that clear? So there is one example. Do you know tadpole, students? Yes, do you know tadpole? So when you people talk about the tadpole, it is the larva of frog. It is the larva of frog. So when you look at the adult frog, that adult is without tail. So what's going to happen to this tail? So students, during metamorphosis, basically when this tadpole is changing its form, when it is getting converted into the frog, so its tail will be eaten up by the lysosome. That's why adult is not having the tail. So that is also the function of lysosome. Is that clear, everybody? That is also the function of the lysosomes. So this is, or enzymes of lysosome, they can digest organelles. So that's why they are disposal unit. So whatever is the product of digestion, students, that will again enter in the cytoplasm. That will again use to form the new molecules. So that's how it is also acting like a recycling center. Okay, recycling center. Is that clear? Is that clear? So next is vacuole. Vacuole, which are very small in animal cell if present, but in fungi and plants, you have large vacuoles. You know that in the case of plants, the central vacuole is very, very, very large, due to which even the nucleus will become eccentric. Due to which even the nucleus will become eccentric. So here what you people are going to see, you people are going to see one thing, that vacuole is having a covering. What do we call it? Tonoplast. What do we call it? Tonoplast. What do we call it? It is tonoplast. It is again an important question. Here also, students, you will see many proton pumps. What you guys are going to see, you people are going to see many proton pumps here, right? So against the concentration gradient also the things will move in the vacuole. So when you are talking about the vacuole, they increases in size, right, as the cell enlarge. I told you this thing already, the water will enter in the cytoplasm, it will also enter here in the vacuole, that will overall result in the enlargement of this plant cell and the vacuole, right, students? So in mature plant cell, you people will see that it is the central vacuole which will fully occupy the cell volume, right? It is the central vacuole which is fully occupied, which is going to fully occupy the cell volume, right, students? So the next part here is membrane that is going to cover the vacuole is called tonoplast, as I said. So it is also selectively permeable, right? It is also selectively permeable. So it is also that maintains the concentration of material different from what we have in the cytoplasm, right? So this tonoplast will help in that. This tonoplast will help in that. And always remember, students, that vacuoles are having the acidic pH. Water will enter in the vacuole through osmosis. So in the cytoplasm, that water will not be there. So water will be demanded from the surrounding cell. That is how the turgidity of the plant cell will be maintained. Okay, so types में we have the sap vacuole, very common type, which is having, free water, which is having variety of compounds, right? We have the contractile vacuole, students, that helps in osmoregulation. I hope you all understand what osmoregulation is. So osmoregulation means maintenance of salt and water balance. What is it? It is the maintenance of salt and water balance. So I'll give you the example here. You people can take the example of amoeba. You know amoeba? Yes. Do you know amoeba? It's a single-celled eukaryote, right? It's a protozoan. It's a single-celled eukaryote, and it's a freshwater organism. So it is living in the fresh water. So of course, students, inside the cytoplasm is there. So we know that it is a freshwater organism. It is having the cytoplasm. So basically, the cytoplasm is more concentrated than the surrounding. So definitely water will enter, water will enter, water will enter. Now, if water will keep on entering it, if endoosmosis will keeps on going, then this cell might burst. And this is not something that is required, of course. So here we have the contractile vacuoles. That contractile vacuole will take up, all that extra salt and water. Then they will fuse with this plasma membrane here, and then they will do the exocytosis of that stuff. So that is how, right? That is how this contractile vacuole, helps in maintaining the salt and water balance. So how? So excess of salt and water, it will be picked up by this contractile vacuole. This will fuse with the plasma membrane here, will throw out all the content outside. So that is how it is going to work. Is that clear? Is that clear? Right? Then the food vacuoles. So they are found in the protozoan protista. They are present in lower animals, right? Food vacuole. Though they are mainly, we discuss it in the case of protozoans. But here you will see many lower animals, right? Many lower animals, my dear students, they are also having the food vacuole. Gas vacuole, which you will see in the prokaryotes, they'll help in students buoyancy. So what is important here regarding gas vacuole? The same thing I have mentioned in the prokaryotic cell, that they are membrane bound. So that membrane is a non-unit lipid membrane. That is not the true lipid membrane that we discuss, but that is proteinaceous structure. So that is present in prokaryotes, help in buoyancy. So vacuoles also help in maintaining turgor pressure of plant cells. So this is all about the vacuole, and this is all about their diagram. Is that clear? Is that clear? So this is something that you all can see here. The nucleus, nuclear envelope, the endoplasmic reticulum, rough and smooth. This is the Golgi apparatus. So transport vesicle from endoplasmic reticulum will fuse to the cis phase. Here the modifications will occur. From trans phase, the vesicles will come out, and that's how it will act. Okay. So this is something, right? This is something which you people need to keep in your mind. Is that clear? Is that clear? So that's it. This is all about today. So we need 2 more classes, students, and in that 2 more classes, we'll be done with the chapter. Okay. And do let me know in the comment section which chapter you want me to start next. Okay. Whatever we are completing or whatever we are discussing here in these chapters, this is something, very important, and there is no need to go beyond this content because I'm providing you everything here. So if you will revise this, if you will read NCERT, if you solve question, trust me, you are going to crack your NEET examination. And if you need more details, right, more help, so you know that, right, the complete Unacademy NEET English team is there for all of you. So you people can watch my recent video, and this is a realistic strategy. If you watch it, you'll be able to understand that what are the things that you people are not doing correctly, and that's why you are lagging in this preparation. So you people can just have a look of it. You guys can watch it. So right here in the description box of this video also, you guys will get the link for the batch as well. Okay. And there is something important for all of you. See this, click on that link, right? So there is some, some surprises coming up for you. So just click on that, just click on this like button, right? And click on the notify me. You'll get to know about the details. And trust me, you are going to find it very amazing. Okay? Done? Okay. So this is all about the today's lecture. So take care, guys. Thank you so much. See you.